Welcome back, everyone, to this first session named Brain Mechanism under Brain Disorders. Welcome back also to Professor Sandi and Dr. Raoul to participate into this debate. So the objective of this discussion could be summarized in 2 words: big picture. It's really about giving to the audience a general perspective of the current view on brain disorders and how you think the field has evolved during your career as a neuroscientist and how you think the field can then move forward. So maybe we can open the debate with a very general question, which would be, since the beginning of your careers, have you noticed a shift in the global perspective around brain disorders in the field?
So they start maybe?
Please.
Yeah, I think it's changed in many different respects. For me, maybe the most noticeable is that many years ago, there was a very important emphasis in understanding mechanisms of a particular disease, let's say depression or anxiety or whatever. And slowly we've started realizing that there is not only one type of depression. There are many different types of depression. So I think being specific, it's very important. And the other aspect that I think it's critical is the acknowledging of individual differences also. So there are many different types of depression. And also when we use models to model diseases, realizing that individual differences is critical to understand the impact of anything, almost any treatment or any modulation, etc. So the change is huge. Now I think the field understands that we should stratify if we want to understand mechanisms, if we want to do treatments. And I think indeed, from my point of view, many of the clinical trials that have failed over the years in the field of brain disorders, and in particular in mental health, it's because there is not a good understanding of all this diversity, but we finally got there. I remember many years ago, not only scientists, which I think reflect the current view of the field, but also the pharmaceutical companies didn't want to hear about individual differences. They didn't want to hear that maybe a treatment could be beneficial to a subset of population, because maybe this was not where the big business was there. And I think this has completely changed because of the failure of the other approach, which is a more global one. So this is maybe where I would like to put the emphasis of this change, because I think indeed, if we maybe use the metaphor of a brain as a machine, it's not a single one. We have to understand each of them, and then also in terms of mechanisms. Of course, there's been also a big progress in the definition of brain disorders by focusing more on the mechanisms, the origins, etc. And then we have the OD approach from the American system that's helping a lot in this direction.
Okay, and you, Cédric, since the beginning of your career, have you noticed a shift in the global perspective on brain disorder?
Working basically on spinal cord, I don't see a massive movement from the spinal cord side. But yes, indeed. Yeah, you saw a lot of things moving, especially as Carmen said about personalized medicine and basically you're gonna stratify patient and a more detailed and accurate classification of patient, which is really important, especially for brain disorders. From the beginning of my career, basically, yes, you saw this is more and more taking into account, and it's a good thing. And because just a kind of a global view, kind of one molecule in terms of therapy, one molecule that will rule out everything, that will cure everything. I think that more and more people basically have that in mind, that is not really, realistic, and that we're going to go through maybe either a complex treatment or basically more specific treatment according to the specificity or the feature of patient. But I'm not much in brain disorder, so I don't have, so much background on that. The spinal cord is more limited. The spinal cord is more simple, and stratification, for sure, it's necessary to have a good, definition and a good idea of the clinical feature of each patient. But I would say that for, more complex disease such as schizophrenia, autism, and all that, it's more important. And you see basically the field moving toward that. And it's very simple to see, how fields moves. You just look at the call for proposal of European Community. You see basically how the grants are now oriented and what kind of goal they want to reach. So you see that it's more and more realistic, and people go toward this direction, which is a good one, I think. Yeah.
Thank you for your inputs. Since you feel like the drive to develop this kind of personalized treatments for brain disorders, why do you think preclinical models are still using animals models of common genetic backgrounds? Why do you think the drive to not have variety in these models is not stronger in the community?
Yeah, thank you for this question. In fact, I've been quite critical over the years on the use of genetically equal individuals, inbred strains of mice or other organisms, because I really think that gene-by-gene interactions and how they also maybe define these gene-by-gene interactions, epigenetic modifications related to different life experiences or treatments, etc., are key. So I also question myself in general if this is something that we should be doing. Finally, even though for many years we've been using outbred animals like oyster rats and other rats to account for this genetic variability, when we want to understand the circuit at the moment, most of the techniques that allow that in a specific manner are developed in the black 6 mice background. So we are using that, but not losing the other model. So we have the rat for outbred, and then we go as far as we can. And then when we need to move to some manipulations that require other definition, then we move to the inbred one. I remember, maybe it's a traumatic memory, 3 or 4 years ago, one of my projects was not approved because I was proposing experiments using rats and mice, and it was particularly for this approach. So I think this doesn't happen now anymore. There was a time when it was even difficult to publish papers if you had also rats and mice, because you were like jumping from the species. And now I think we see that this is an added value also to use to have human data, etc. So yeah, what to think about it? So the way I think about it is that we shouldn't forget that when we use black 6 mice, it's like one single individual from a genotypic point of view. So many times, in fact, when we have findings in rats from outbred strain, and then we move to mice, we see maybe differences, but not going in the same way. And I'm not too scared about it, because not all the individuals of an outbred strain show always the same effects. Let's imagine effects of stress. There is variation. And then when we pick up one of these genetically equal strains, strains, then it's like one individual from a genotypic point of view. So the way for me to use it is to understand in that particular model how variations on a particular aspect affect the function of the circuit, etc. But really, afterwards, I like to go to the real, maybe a genetic variation, and see how it accounts. So it's not a simple answer, because for me, this is a very critical point. I'm a bit worried to do so much work on a single genotype, and this is too much work that we're doing. So I think we have to find ways to overcome this bias from some point of view. Thank
you very much for your answer. Yes. Maybe we can ask the same question to Dr. Raoul now?
No, it's right. I mean, it's completely right. It's very frustrating because you have one gene on one inbred strain, and it's one aspect of the disease. And sometimes you've got the feeling that you just work on some mouse specificity that has no relevance in humans. And there is some, in my case, in motor neuron disease, that you see some things that it seems to be idiosyncratic to it's really specific to mouse, that you don't retrieve any sign in humans. So basically, it could be really frustrating and you questioning about your role as a biologist. I mean, if you just work on the biology of mice, it's interesting for sure, but you need also applications. So, but it's right that we are limited in terms of model. We don't have much. And we just hope that what we identified in mice or rat is basically one common mechanism that you can retrieve in mice. And regarding the complexity of the human situation with environment interaction and so on, I guess that with mice, we have a lot of targets, possible pathway, which we describe, I mean, not only in neurodegenerative disease, but in brain disorder and dysfunction, you have a lot of targets. So if you have a lot of panel of pathway and drug, according to what you can identify in patient or which type of patient, which specifically may, some activation of this pathway or another, you can maybe play with drug and adjust the panel of drug that you have. This is a kind, I don't know if it's really true, but it's working with one animal model, it's really frustrating. So the only way you can do is to basically combine different genetic model that leads to the same disease. So it's one aspect that if you have, what we work, for instance, in SOD1, if you retrieve the same in TDP43 mice, in first mice, it's more and more, you seem to trust at the end your results in terms of clinical application. So it's working with outbred, or it would be great actually to combine one genetic mutation in different strain. But the point is a place, and it's not possible. It's not, especially now, it's more and more complicated to do animal experimentation. But the idea will be to indeed take one genetic mutation that causes a disease and explore interaction in different strain of mice and so on. So it's one also possibility, which is very space consuming. It's a very cost effective actually.
Okay, we have question from the chat. How will you design a perfect experiment in rodents to implement the paradigm of personalized medicine, provided that your resources were not a limiting factor? Which parameters will you vary? Which biomarker will you use?
Okay, this sounds like a dream project full of resources. I think indeed something that I would do if that was possible is to start working with a naturalistic environment with a large colony of animals in which I could follow every animal with different approaches. Now there are many in which we can follow all the behaviors, interactions, and also, if possible, also metabolically. I think the question relates to looking for biomarkers right now because of the work and how the field is evolving. I think one of the key questions to understand also personalized medicine, but it's to understand the origins of individualization. What does it make us different? So of course you can start with genetic variability, but you could also have colonies in which all the individuals are genetically equal, and then still you have the development of individual differences. And this is something that one could put together all these different data, big data, and then eventually see how the different phenotypes evolve. Of course, we would need to give challenges in the field of brain disorders. So that would depend on the question. If I'm thinking about mental health and more my field related to how stress-related disorders emerge, it's quite complex indeed because life can have many different types of challenges, and then it depends on whether we want to understand early life challenges or life events, etc. But I think, yeah, you get a little bit the picture. I think by getting a lot of data, now I am very excited about how much we can get also from behavioral data of very rich, high density data that eventually we can map onto disorders, if you want, when you have a big population and one can impinge different types of challenges like diminish the resources so that individuals have to compete, but also this brings scarcity, and this brings the need of individuals to fight. So this would bring some chronic stress situations, etc. Then one needs to map it. So I'm not sure if I answered the question, but if I would have a lot of resources, I would like to tackle both genetic variability versus non-variability, so that I could see indeed how the individuality emerges in both cases, so that we can account for the importance of the genetic background. Because I think something that it's amazing is indeed that I was criticizing the use of inbred animals in the previous question. But what is incredible is that even when you have inbred animals and you put them in a colony, you get a full spectrum of different behaviors and also vulnerabilities. So there is something on both how the environment affects the individual, but also I think very important that I think it's something maybe to discuss a bit further, is the social environment. How individuals relate to others has a huge impact in the mental health and also in the physical health, that somehow we are not yet there in capturing this aspect. So to summarize, I would do this gigantic experiment in which I would include the perturbations, and I would be very interested in looking at behavior, what does it tell us, and also as biomarkers metabolism.
Okay, and you, Cédric?
Yeah, it's quite tough. Question is really tough. I would say exactly the same. It's kind of giant experiments where you combine with one mutation or two, one or two mutation, and just express them in different genetic background and expose your animals to a different, environment. But it means a lot of space, it's a very giant experiment in terms of follow-up. And the problem here, the question is that the term of the question are, paradoxal, because the principle of individuality is basically not reproducibility. It's not reproducible. Individual is one thing that happened once. And the problem in science is that if that happened once, it's not good. You want to reproduce and to repeat. So once you start to repeat, you lose the individuality feature. So it means the only way to do that is to have a lot of individuals, and indeed after process all the data and try to identify some things mathematically, I guess. But the term of the question is paradoxal for me. It's reproducibility and individuality. I have no idea. I hope, yeah. Except a huge experiments where you combine, you've got a huge farm in the middle of nowhere. You have got 3,000 mice that you can explore with a different genetic environment and through different and a lot of battery of behavior and so on. But it's a giant project. It's huge.
Like, would you go for super big and performant computers and big analysis, or would you stay on?
Yeah, I would say that, because basically the principle of inbreeding is that you've got one point mutation in a very controlled genetic environment. And so basically you can reproduce. It's what we saw in motor neuron disease. There is no surprise. Honestly, we've got the mice we have. I don't think it's the same for other genetic disorder. But in our case, it's a Swiss clock. The mice at 5 days, we know that she's going to start by the same population of motor neurons in the same region of the spinal cord. Then after, we know that this marker will be activated, and so on and so on and so on. we've got a really, really detailed analysis. But this detailed analysis is feasible because it's inbred with one genetic and everything is controlled. So it means that when you start to, put inside something chaotic, something different, it means that you're going to have a lot of variability. So variability means increase the number to extract something from that. So it means mathematics, basically. I don't say modeling. Oh yeah, you will have to modeling to big data. It can be a big approach, but it's an approach, but it will be approached through mathematical treatment of the data, just because the limitation of this variability of individuality. It's not a definite answer, I know, but.
Thank you for your answer. We just received another question. Are there ways in which citizen science does support or could support research of brain disorders?
What's the question? Citizens in science?
Are there ways in which citizens science does support or could support research of brain disorders? I'm not sure to understand the questions myself.
Yeah, I think it's non-scientific, like basic
Yes, maybe,
citizen can support brain research. Is there a way when you are not a scientist?
Well, if you are wealthy, you can support maybe with some sponsorship. Of course, if that's not the case, still, donations can be one way. The other way is participating if you are one of the individuals that fit in particular cohorts. I think nowadays there are many also online platforms in which people can participate in experiments, etc. Sometimes I receive emails from people that suffer a lot of anxiety or stress or depression, and they want us to study them in the lab. And of course, this is not something that we can do because we need to have a very specific type of recruitment, etc. So I think it's very important indeed that people are motivated and non-scientists to contribute. I think we scientists have a lot more to do than what we are currently doing in outreaching, and then maybe from this dialogue in this we can all learn. I think in some fields there is much more interaction with patient organizations and also with the general population regarding specific disease that maybe are less stigmatized. In mental health, we are not so much yet there, but I think, now with all these online opportunities, probably it's something that we should start using more in order to connect with the public. I think indeed by being well informed is already a very important way to contribute, because there is a lot of fake news and a lot of, I think, not clear attitudes towards mental health or brain disorders, etc. And by being well informed, people can also already contribute a lot, I think, to the advancement of science.
Okay, apparently the person that asked is bringing some more. So citizen science is collection of large population data starting from 10,000 persons for e.g. occurrence of certain symptoms through questionnaire in order to narrow down the research.
Yes, I think it's just some details about the questions we just answered. But maybe we would have a question in this aspect with the relationship to the general public. So lifestyle choices, diet, cognitive exercise have been shown to influence brain health, of course. But it's sometimes difficult to the general audience to understand that, especially when they don't show any symptoms. So would you have any recommendation on how politics in general could then guide and improve the promotion of these guidelines to the public?
Cédric, would you like to answer?
Yes, I can. I would say basically through, the most important action is education, basically, first, at school. I think it starts early. it's very important that to act at a very early in the process of education. Where you can start basically at the school restaurant, where basically you start to educate. Young people, explaining that basically it's bad to eat. It's better to eat a green peas compared to French fries full of oil and so on. So it's very stupid, but there is more and more. People are conscious about that. So at least in France, I think it's the same in a lot of countries. But they start to be really careful about what young people have, and they explain. So you've got, a role of really explaining and try to educate people and explain them that it's good for health, it's good for mental health, and so on. So it's I would say basically it's not very original answer, but education very early, it's really important. And also consider that this is really important throughout the world. Our life is not only when you are 3 or 4, but it's also very important when you are 40, 50, and more when you are 80 or 90. But I think it's a matter of advertisements, communication in the media, in the social network, and after also on the lobby on the market, basically try to basically fight junk food and try to basically help people to do not go through the easy solution to eat hamburger or whatever. That's sometimes good life requires effort to prepare things, but it's worth doing it.
Perhaps I can add something here, if it's a little bit out from our current experience. I think another way that governments can help is by having scientists also communicating to the general population. We've seen now that with the COVID pandemic, it's been very important that scientists were communicating for credibility, etc., because otherwise everything gets politicized. And one way, I think I'm happy that we have this chance, this opportunity in Switzerland, that there are projects that we can propose for public outreach, and we just got one to bring to the general public the field of research on stress. And we are going to organize a very, I think, a nice website, also days, research days, people can visit the labs, etc. So I think by having the scientists being able to popularize, to bring the science to the general public is one way in which many of these new measurements can be implemented or can be better explained. So this is my proposal. I think we have to cooperate a little bit more.
You're right. It's indeed important, but it's what I saw in France, especially with the COVID period, that there was a lot. We had problems with, scientific and clinicians that claim a lot of things that at the end we showed it was wrong and the data manipulated and so on. So on the other hand, it's really bad because, the audience considers sometimes scientific, people like not really, they don't trust as they should trust us, or at least clinicians. So it's also the function of politics to basically take care about integrity of people they speak, and to who you're going to give a lot of importance when they give advice. So it's also important. We've got this crisis in France with some. Sometimes scientific say something, and the day after you've got another one says the opposite. So it's not really constructive. And at the end, it's not, positive for the large audience. They just think that we don't know what we are doing, basically. So I don't include myself, not clinician, but it's important too.
Okay, well, thank you, both of you, for participating to our symposium and panel discussion. It was really fascinating. Thank you a lot.
Thank you.
Thanks to all of you.
Thank you.